it's Dr. Oi.
And I'm Freya. You're watching Animals Speak, and we are here at the National Laboratory Animal Center of the Mahidol University.
That's right. Today, Freya and I will introduce you and all of our wonderful viewers to the small animals that benefit mankind. Explore the world
Of animals
with Animal Speak. Hello.
Hello.
Freya, this is Director Kanchana.
Hello.
And this is Dr. Pradon. I told Freya that we're coming to see the small animals in the center.
Why do we need laboratory animals?
These animals play a very, very important role in our lives for finding cures for human diseases. They benefit humans a lot. They have been used for medical experiments to produce medicines for humans. They can also help humans in pharmaceutical testing, research, or body system study. We've learned a lot about medical treatments for humans from them, and of course the treatments of diseases from these particular animals. What is the purpose for this lab and when did it all begin? Well, before 1967, we needed to find rats and could not find any, so we bought rats and mice from the street markets along the road. But they had low quality and suffered diseases, which could be passed on to humans too, and they kept dying in our care. Finally, in cooperation with Chulalongkorn University, Kasetsat University, and Mahidol University, the Standard Laboratory Center was established with a joint agreement to foster quality lab animals.
What kinds of animals are there in this center?
We initiated with rats.
Big white rat.
We have mice too.
Oh, they're a little bit smaller.
Rabbits as well. And also guinea pigs. All four kinds are called lab animals because their purpose for scientific experiments and research.
Those lab animals are different from normal animals, even though they may just be a regular rat, a mouse, or a rabbit. They do have a very high value here, though. Each one will be guaranteed by the experts to be of the highest quality to comply with the standards, and each one has to be void of any diseases. And we usually test their quality of each animal before sending them to our customers.
Those being used in experiments are raised under quality standards and healthy environments. We also bathe them before putting them into any experiments. Wow, they take a bath. Freya, did you take one yet?
Yes, I did.
Well, even if you already have taken a bath, you still need to take a bath when you get here, and you need to change your clothes as well.
Are you ready to go inside,
Yes,
Freya?
I am.
Okay, I'll show you how we grow these lab animals with the highest quality.
So we'll say goodbye to Dr. Pradon right here.
Bye. What are these sacks?
Oh, they're animal food sacks. We'll receive more mouse and rat food today. They will be sprayed with a 70% alcohol solution to sterilize the sacks as much as possible before keeping them in a storeroom. Freya, do you know what these boxes are used for?
They're probably for containing something.
It's used to contain those mice before sending them to our customers. You see the cover and widescreen over here? It can open and it can close. Mice have very, very sharp teeth, and that's why we've designed a special box for them to prevent gnawing. And there are breeding vents here also, just to follow the regulations of the Animal Transportation Association, or ATA.
What are these used for?
These are bedding materials. The soft bedding materials are used for sleeping and to absorb their waste. Actually, mice are very, very clean animals. People often think that mice are dirty animals, but actually, they're the clean ones. They clearly divide their space into separate parts.
They divide into sleep zones and urinating zones.
Exactly. These are important materials because animals will have direct contact with them. So, there is a whole sterilization process which they will need to go through before use. They will be steamed before sterilizing, and after that, they will be labeled with a sticker with the date in which they were steamed, and also the expiration date within 7 days to make sure that the sterilization is still effective.
Freya, do you see this tape? It used to be this color. Now, this is used to indicate the temperature. We will attach it to the sack. If the temperature reaches 121 degrees Celsius, a black color will appear. It means this sack is sterilized.
Freya, we're now at the washing zone. Do you see this? This is called a conveyor belt. It's used to transport the used cages. The used cages will be sent to the cage washers. We have 2 machines now. All animal equipment must be cleaned and sterilized before use. And these are the cage covers. They will each be washed to make sure the cover is cleaned.
I want to see what's inside. Do me a favor, Freya. Can you check it out for me?
No. Of course not. That's impossible. You cannot go in there. There's the cold water, the disinfectant, and the boiling water. After that, the equipment will be set to 92 degrees Celsius heat. So if you get in there, you'll be well done.
Oh, and ready to be served as pot roast.
After the washing and drying are finished, all cages will complete the steaming process. This is the 2-door autoclave, which consists of the dirty and the clean side. This one, in the short sleeve shirt, is working on the dirty cage, while the other, in the sterile suit, will be at the other side of the machine to work on the clean one. There is also some equipment that cannot be used in heat, so it'll be put in this room. It's the decontamination chamber with the controller outside. The machine will emit gas to sterilize the equipment. And after that's finished, the equipment can be brought out from the opposite door. As you see, all the equipment used in the feeding room will be cleaned and sterilized before use every single time, before usage. Everything needs to be cleaned. Yeah, and sterilized over and over again, just to make sure.
Oh, wow! What is this? Is it a giant strainer?
A giant strainer?
Yes.
No, it's not a giant strainer. It's a mouse strainer.
Yes, it's used to filter water for the mice. They have to drink clean water filtered from those strainers by a reverse osmosis system. Have you ever heard of reverse osmosis? Yeah, it's very popular, especially among human beings. But who would have known that these mice have been drinking RO water for a while now? Well, the last one we saw was the first filter process. This is the second filter process, used to remove scents from the water. And then the water will be released into the main drain. The most important process is to clean up the water. The water will be maintained in this tank. Water in this tank will be circulated all the time. Otherwise, it will be the origin of bacteria, or what we call dead space.
Did you know that mice in this laboratory center drinks over 1,000 liters of water per day, and the whole center consumes about 30,000 liters per day?
Okay, Freya, we're going to see how they care for the lab animals. This is Dr. Surachai.
Hello, Dr. Surachai.
Hello.
He takes care of using the lab animals in the experiments, and he'll show you both how they work, okay?
So, Freya and I will go with Dr. Surachai for now, and we'll be back with you again, Director.
Okay, we'll see you later.
Before entering this room, we have to take a bath and change into sterile suits for cleanliness.
Okay, we are now inside, and this is the sterilization zone, which has very clean and sterile equipment. Our staff has allocated them into separate lots here, ready for use. The next room is the food and bedding storeroom. The temperature here is quite low in order to preserve food so that the food does not go to waste. The temperature here, we usually keep it at around 21 degrees Celsius.
Uh-huh.
The next room is the water preparation room. The water preparation room has filtered water from the RO system, which will be sent through the stainless steel tubes from the water generator. The tubes run parallel so that there is a circulation process to prevent the dead space. After that, the staff will add chlorine into the water with a 3 to 5 ppm concentration of the solution.
It's one of the sterilization procedures, right?
Yes, to prevent infection when they drink the water. The next room is the waiting room.
What is the waiting room?
The production zone will send animals from the other side or the dirty side to be ready for experiments. The boxes of animals will be sent through this door. The staff will then spray disinfectants around the boxes and move the animals from the boxes to the cages. Those animals will stay in this room for 1 day to check their health before being moved to the feeding room. If they're all confirmed to be in good condition, they can be moved to the feeding room.
Okay, let's see where those mice are going.
Yes.
Wow, their sleeping room is so big.
Right.
Yes, it is. After we move them here, they will be moved from the old cages into new cages,
Uh-huh.
weighing them and feeding them food and water, rechecking their health in detail, whether they have any abnormalities or not.
Did you know that there are about 600,000 lab animals used each year? However, this laboratory center can produce 800,000 lab animals each year. The care of animals at this National Laboratory Center consists of 2 levels, a low barrier level and maximum barrier. The difference is the maximum barrier level is safer than the lower barrier.
This room is the washing room for all the used equipment. The equipment will be prepared for cleaning and put on the shelves. This shelf has all the cages on it. As you can see, we have the bottom of the cage on top and the steel on the bottom. This shelf has a very special feature called spring arm
Oh, the spring arm.
to help cleaning inside the cages, while those spring arms help cleaning outside the cages.
Uh-huh.
The washing methods have 2 ways. They are using alkali, then the acid,
Ah.
and cleaning water before blowing with 110 degrees Celsius temperature.
Dr. Surachai, are those the complete tasks within your duties?
Yes, it is, and also within my study.
I also think that Director Kanchana is waiting to show us more in another zone of the center. Thank you so much for showing us around today. Goodbye.
Goodbye.
Bye.
The next zone, we're going to this laboratory zone. which is used to check the lab animals' standards, with certification from many institutes to make sure our lab animals and staff here are both standardized and certified. Now, what are we going to see first?
Well, what do we do with the lab animals after we're finished using them in the lab and moving to the quality checking room? How do we divide them into each pathway?
Freya, take a look which path is allowed to minimize contamination. Now, we must walk from the cleanest area into the dirtiest area, and we're not allowed to walk from the dirty area into the clean area to minimize contamination.
That's right.
So, get that?
Yes.
What is this?
Well, Freya, what do you think it is?
The emergency shower.
Want one, Freya?
Yay!
Well, when you pull the yellow handle, it releases the water for showering your body in case of emergency contact with chemical substances. This emergency shower will help clean the body immediately.
Oh, there's a drain over here.
Well, the best way is to try not to use it, because if we use it, it means we've been in contact with chemicals. Before going into the lab, we have to change our clothes and put on gloves. They are used for blood collecting and collecting organ samples for uses in the experiments and tests by qualified expert scientists.
After the separation of the biopsies are complete, they'll be weighed to distinguish the testing results. After that, those biopsies will be soaked in the formaldehyde to distinguish their condition, whether they're normal or abnormal.
So tell us, how long does it take for the soaking to complete?
Well, it takes 48 hours to change formaldehyde, and they will be soaking until there are enough biopsy numbers for testing.
And moreover, the concentration of solutions can be increased and changed from formaldehyde into alcohol. Is that true?
That's right.
After increasing the concentration by adding alcohol, lastly, those biopsies will be put into paraffin and molded into blocks.
Yes, that's right.
And what is this?
Try guessing what it is. What kind of organ is it?
What kind of organ is this?
It's a liver.
This is a liver of a mouse inside the paraffin. It was sliced into very thin slices.
Yes, and they were dyed before studying the results.
This one is a kidney. Let's watch it through the microscope. This is a kidney, right?
How can Freya see? Pick her up.
Oh, what have you been eating?
You haven't even picked me up yet.
Well, if you look here, Freya, you can see the red color pallets. They look like something round. It's called the nephron. Those nephrons are invisible to the naked eye. If we want to find an abnormality, we have to observe the bleeding, the infection, or bacteria through the microscope. We couldn't tell without these scientific processes. Freya and I are done with our tasks for today at the National Laboratory Animal Center. Freya, what can you tell us about our tour today?
Well, I was really excited about coming here today. I learned a lot about those small, wonderful animals. They can help us a lot in medical testing. Like when there is a drug being researched, these lab animals will help us with the results of the drug's effectiveness. They sacrifice their lives for us humans.
As the director of the National Laboratory Animal Center, do you have anything to tell our viewers?
Well, working with lab animals may sound a little cruel, but actually, they provide a wealth of knowledge for human development.
Oh.
Imagine if we didn't have animals to test with, then we wouldn't be able to test drugs that we were experimenting with.
That's right.
We wouldn't be able to develop our lives without taking any risks. Researchers don't consider them as just mere animals, but we think of them as living, precious things. We think of them as living, precious things because they have contributed so much for the well-being of humans. They have made our lives better.
And for that, we are thankful. As for today, Freya and I have to thank you, Director Kanchana, for letting us visit the National Laboratory Animal Center. And we've learned a lot today. Our time is up today, and it's time to say goodbye. See you next time in Animal Speak, where we will explore the world of animals. Goodbye! Bye.